Hi, so my name is Professor Brouzes. I'm Associate Professor in the Department of Biomedical Engineering at Stony Brook University. So first I want to thank the organizers for the opportunity to present some of our work today. And my goal here is twofold. First, I want to tell you about the story of this project that led us from single-cell encapsulation to digital PCR platform that could be applied to detect SARS-CoV-2. And really, the second aim is really to share my enthusiasm for microfluidics and to try to convey and highlight the creativity involved in the development of microfluidic systems. So Stony Brook University is located on Long Island, close to New York City, and you can see we got like about 26,000 students, including like 17,000 undergrads and about 1,500 faculties. The Biomedical Engineering Department is really a joint department between the College of Engineering and Applied Sciences and the Department of Stony Brook Medicine. So my lab is called Microfluidics for Quantitative and Genomic Biology. What we have been developing in the past few years is really a way, we really focus on how to manipulate a single particle or single cells in a very passive way. The impetus for that is really to develop new systems to analyze single cells, but really single cell at a time. So in conventional methods, like you can see here, when you measure a parameter across a whole cell population, what you obtain is the average measurement. While if you can measure the same parameter cell by cell across that population, what you get is the distribution of this parameter across the cell population. And there's a lot of application where that type of information is critical. One simple example is cancer, where one bad apple, if we can say so, one cancer cells can give rise to a tumor. So that is really important to get this fine description of cell subpopulation. It's also true in developmental biology, in brain science, and so on and so forth. Now, what we're interested in really is we are more interested in genomic analysis of single cells. And my technology of choice is really droplet microfluidics that I contributed to develop while I was working for RainDance Technologies. In this technology, samples are partitioned in small droplets and routed and manipulated through microfluidic channels at very high throughput. Right, so this technology is typically hailed as high throughput or very high throughput technology because droplets can be generated and manipulated at thousands of droplets per second. Now, when I went back to academia, I looked at the single-cell genomics field and noticed the discrepancy between the throughput of droplet microfluidics and the capacity of sequencers. There is a few thousands of single cells per run, which is still the case today. So I decided to focus on what I called pressured samples, such as biopsies, and retool droplet microfluidics for the purpose of analyzing this type of samples, where with the idea is to trade throughput for higher degree of control. And the first thing to basically, the first aspect to address is the fact that single cell encapsulation is limited by Poisson statistics. One thing I haven't mentioned is like really droplet microfluidics is really a very attractive way to manipulate samples through microfluidic channels because there is no dispersion. All the content is maintained within that droplet, right? So again, single cell encapsulation is one of the limitations because it's dictated or limited by Poisson statistics. If you see in this video, single cells come into the nozzle, get encapsulated, but the true statistics within the droplets is what you see on the left, bottom left side, right, graph. And that graph corresponds to 1 cell per droplet. Right, the concentration of cells coming in is 1 cell per droplet. But what you get in reality is the distribution you see in orange at the bottom left here, is that 1/3 of the drops are empty. 1/3 contains one single cell, and the rest of the droplets contain more than one single cell, right? Now, if you want to have true single cell encapsulation, or at least single cell encapsulation, you have to dilute your sample at least tenfold, and you obtain the blue distribution, where all the droplets that contain a cell contain only a single cell, but then you got 90% of the droplets that remain empty. And that's one of the big limitations. So to address this limitation, we developed what we call the trapping and encapsulation strategy. So the first step, we trap single cells into chambers. Then the second step is to flow oil around those chambers. And we're going to see in details how that works to create 3 droplets, right? We've got a amount of a volume of liquid here, of aqueous liquid, basically immersed within oil, so that's a droplet. And the third step was to retrieve those droplets by flowing oil from the other side. So the first step is really to trap. So how does that work? When you've got, you have to consider the module here. You've got the chamber. You see that it's connected to the main channel through a very small channel. And the whole idea is that when the cell comes in, and the device, it's designed so that the next cell comes in, it's going to plug that small channel, right? So that there is no flow through that chamber, and the following cell is going to go around. That's what you see in this schematic. So this is a theory that's designed for, but the question is like, what's the efficiency of this approach? And in other words, if we inject 100 cells, how many can we trap? And here we can see a time lapse that this basically highlights the trapping trajectories. We can see in the first panel on the left, the cell comes in the first chamber. And the second channel, the second cell comes around the first chamber and get trapped into the second channel. And again, in the third channel, the third cell goes around the first chamber, goes around the second, and gets into the third channel. So that's really the principle at work here. So, but again, how many cells can we trap? You can see the video here that displays exactly the same idea. Here we focus on the next empty chamber and the next one, and so on and so forth. And that's really how we quantified. So we looked at the first empty chamber and we looked if the next cell would get in or whether it would get in the next second to next, right? So, and what we observed is like we've got almost 94% of the cells would go into next available trap or chamber, and about 5.6% would go to the next plus one, okay? And the rest would go next plus one plus one. So the trapping efficiency is very, very good. We can say it's 100%. As you can see in this video here. So now the encapsulation. The encapsulation really harness the idea that the cell plugs this small channel, right? And when we push oil, the oil cannot go through the chamber, but it has to go around as well. So the result was not as good as trapping, but still 86% of single cells were trapped when they were fixed cells, while 78% when they were live cells. Now, the third step is really droplet retrieval. By injecting oil from the outlet, we can retrieve those droplets containing single cells, bring them to other modules to process their content. So the take-home message of this project, that was the first design that was published a few years ago, is really it's a passive method, right? It's all like all the cells are manipulated through basically flow. It's all passive. There's no like actuators, electric field, nothing, just flow. We don't get any empty droplets per principle. The droplet volume does not depend on cell size, but on the volume of the chamber itself. And once trapped, cells can be observed, right? And our idea is really to combine those observations with some genomic profile and perform like multi-dimensional analysis of single cells. Now, what can we learn from encapsulation failures? So here, this is a time lapse, right? In this column from top to bottom, it's over time, and the oil goes from top to bottom as well. So in the very top panel, what we see is like the oil coming in, and you got this interface. So the interface is going to snap off, but if you observe really what's happening with the cell, you can see that it gets squeezed in through the channel, the very thin channel, and get expelled through it. Once it's expelled, basically all the content of the that is in the chamber is going to be expelled as well. So that's how it fails. And basically, what happens is like here in the schematic, again, the oil comes in, create a higher pressure that is going to create some flow around the cell if there's a little bit of leakage, and that is going to deform the cell and push it out. So what we realized really when it's basically efficient or like effective is that the encapsulation is complete when the front of oil reaches that small channel here and cuts it off. We realized that, okay, after that, the system inside the chamber is refrozen. And our idea was, okay, why don't we then cut off this channel first? In other words, why can't we inject the oil from the outlet, cut off the small capillary channel, and go around and complete the encapsulation process? So the oil, we can cut off the small capillary channel without getting in thanks to capillary valving. Now, there's an issue with that approach. It's called full reversibility, right? We push the cells in like this, it gets trapped. But now if we push oil from the outlet, we're going to push the cells out again. So again, our first naive idea was, okay, let's create a secondary capture, putting a hook on the way out. Now, if we really look at the trapping principle here, is really to understand how things work and how we can really address all those issues at once. So here you've got the chamber. We cut off basically the loop channel here, but the flow goes around and goes back down, right? And what happens, like part of the flow goes around the loop channel and part of the flow goes through the chamber in red here and blue for the loop channel. So what happens is like if you want to be able to trap a particle or a cell, the diameter of that particle needs to be within the rate flow. Okay, like depicted here. That's why we developed those zigzag structures that we could see on the previous videos. So now if you put a hook, this is the problem here in the first panel. When you put a hook, that you're going to prevent what we call the primary trapping, right? No cell would go in. Now if they can get in, you've got perfect secondary trapping. Now, if you open up a little bit more or too much, you may get 100% primary trapping, but 0% secondary trapping, right? So there is a way to get a little bit in between, not 100% primary trapping and not 0% secondary trapping. That's quite inefficient. Our solution was the following. Basically, we add a pinball system with the hook. So the cells get in, get trapped at 100%, and then when it's way out, it gets deflected at this new structure inside the chamber towards the hook. And basically, one of the big ideas here is like we break the flow reversibility by inducing cell structure interactions. So let's see how it works in practice. So what you can see in the video, we've got a bead here, which is about 16 micron in diameter, gets in. You can see the primary trapping. It goes around the hook, around the second structure now, and gets trapped. Now, when we reverse the flow, here by reversing not just with oil, actually, with air, the particle gets dislodged and pushed towards the hook. And this approach opens up a new possibility by using a tertiary trapping by the small hook here. Now, the initial particle is trapped, so we can bring a second particle in the system. We get like trapped into the primary trapping. And when we reverse the flow, basically pushed into the hook as well. Actually, more a hook here. So you can see by developing this new design, we can do perfect single cell trapping, but even perfect cell-cell pairing or cell-particle pairing, which is really like elusive goal in microfluidic world. So we can see here at work, secondary trapping. That means like the first particle, right? Again, we try to quantify as much as possible all the steps. So we got about over 90% success. And now we got the pairing, the second particle now, which is a cell in white. It's difficult perhaps to see in the video here, but they're getting in. And we got like over 92% of trapping the second particle, or cell in this case. So really in terms of principles, we broke the flow reversibility thanks to cell wall interactions, and in different ways with cell displacement structures. These are the zigzag structures, and you could see in the different videos, and the pinball structure inside the chamber. That will really like enable new possibilities here, right? We can do perfect deterministic cell bit pairing, cell-cell pairing, particle-particle pairing, you name it. Now, the next steps we're working on right now is to show usefulness of this approach to perform single-cell cytokine real-time detection and single-cell genomics and epigenomics. And again, at the very high efficiency, geared towards like pressure samples that contain between a few hundreds, typically a few hundreds single cells. Now, I'm going to switch here to, again, switch and go back in time, right? So go back to the basic principles and see what other opportunities we could find here. So why, I mean, basically, why does it work? How is it possible that we can encapsulate from the outlet, right? And just focusing on this small channel here. And I mentioned already that the fact that we can inject actually oil or air through the outlet and encapsulate, it's because the second phase, right, the air or oil is not going to go through the small capillary channel because of capillary valving, okay? And that is due to Laplace pressure. So you got this interface. It doesn't want to get into a very small opening. It costs too much energy, so that's why it goes around the loop channel here. Is going to close, encapsulate that chamber from the other side. Now, what are the new opportunities here? It turns out that you don't need a plug. You don't need a cell, actually, right? To be able to perform an encapsulation. So in this case, we can create empty droplets. So it's not necessarily a good thing for single-cell genomics, but it opens up new possibilities. Because if you can create a droplet like this, that means you can create a lot of them, and you can perform digital PCR digital assays, right? So that's basically a new way to perform droplet generation. And the new way, we're going to see what it means and what are basically one of the main problems in microfluidic in general, especially using arrays of chambers. This is the following. We've got a bottle here, a very simple bottle, and we try with a funnel to fill that bottle. And what you can see is that basically you cannot fill a bottle or empty container like this. Now, if you put a straw here, now it's easy to fill the bottle. And the reason is very simple. Because your empty container is not empty, it contains air. That needs to be displaced to be replaced by water. So without the straw, the air cannot be displaced. There is no escape route. Now with the straw, that creates an escape route, and then you can fill the bottle. So filling an empty container, right? Again, it's like empty containers are not empty, but filled with air, and there needs to be an escape route for the air. So in microfluidic devices, that challenge is resolved by using mostly PDMS because it's gas permeable. So the escape route is the material itself. But now PDMS is not cost effective to large scale manufacturing. On the other hand, thermoplastic is great because it's cost effective for large scale manufacturing, but it's gas impermeable, right? So, and basically you see, and the question is like, how can we use thermoplastic to recreate partitions at large scale? And the answer is like to use capillary valving to control liquid air interface during device filling. And this is the type of chamber we've been using. You got the main channel, horizontal here, and you got this loop here, the chamber that has a large opening and that ends up with a few capillary channels that are very thin. So what happens, the idea is like we fill with, let's say, a liquid water in this case, right? The interface, because we've got an interface between liquid and air, comes to the opening of the chamber, and it's going to get into the chamber because it has a much wider opening than the main channel. Now it can get into the chamber because we created escape route for the air through the capillary channels. And the interface progresses until the end, until it reaches the capillary channels. At this point, the interface will go back progressing through the main channel. Now, once you've got that, you can inject oil from the outlet, and based on the same principle, it's going to go basically stay within the main channel, won't go through the capillary channels, and create a partition. Now, that is at low capillary number because we harness capillary effects, but we can use the same structure at high capillary number from the inlet. And in this case, the progress of the ore is going to depend on the hydrodynamic resistance. So you have to make sure that your hydrodynamic resistance of the main channel is lower or much lower than the ones through your chamber. And these are two modes of performing the full encapsulation. So now we've partitioned an array of 4,800 chambers, and what we basically obtain, take-home message here, is that we got a standard deviation that is lower than 10% in terms of volume. These are still primary results, but still it's good because we need less than 10%, we know, for having accurate quantification. So this opens up new perspective, right? Because we can make those devices in COC imprinted with soft tool. We bonded them with pressure sensitive tape that is very easy to manipulate, which is really interesting. Like, it's quickly deployable, but in addition, it's functionalizable platform by spotting. What I mean by this is that because we use pressure sensitive tape, we don't bond, we don't close the channels with any thermal treatment or any solvent that could damage anything by a spot with a microarray spotter, such as nucleic acids or enzymes or proteins. So it really opens up a lot of applications, and we are working on this to make it more robust and basically make it like equipment free. So just to recapitulate our capillary journey, we started with deterministic single cell encapsulation, and we realized we could actually do deterministic particle pairing and encapsulation. And now we exploited that further to develop some digital microfluidic platform. So now I would like to thank people really like develop and delivered all this work. For single cell encapsulation, we got Doctors Sozaid and Christian, and for digital assay platform in thermoplastic, Dr. Kwan and Mrs. Maria Alvarez Amador and Yue Yang, as well as our basically funding agencies. And thank you very much for your attention. I'll be around to take any questions.